All right, back at you with the second video for today. So in this case, or in this video, what I want to run you through is kind of a real-life example of why alternative splicing is important. So remember back to the first video, one of the key functions of alternative splicing is to actually regulate gene function, right? So if you include or exclude different parts of a protein that are coded for by a gene, you essentially produce two different proteins, and these two proteins, or more depending on how many splice forms there are, can have different functions, right? So depending on which splice form is used, that can then regulate a protein, which then can affect a cellular process. So by extension, alternative splicing itself is actually a pretty key, important regulatory mechanism to regulate different cellular processes. All right, so in this example, we're going to be talking about this gene right here, BCL2L1, or BCL2-like 1. So here's the gene, BCL2L1. This ends up coding for the protein BCLX. As we go through this, you'll see that there are two main splice forms for this one. We'll run through that here in a second, right? But first, a little more broad context. What is regulated by BCLX, right? So BCLX is a protein that regulates a process called apoptosis, which is the fancy word for programmed cell death. All right, so in multicellular organisms, it's actually really important for some cells that they die at the right time. This could be as a regular part of development, or it could be in response to other kinds of cell death signals, like too much DNA damage or too much other kinds of damage caused by stress in the cell. It might be a good decision for that cell to actually undergo death rather than try to continue to survive. That process is called apoptosis, i.e. programmed cell death. Very complicated topic, very interesting. A lot of it kind of really centers around the mitochondria. So that's why I've drawn up this diagram of the mitochondria right here. And what I'm doing is I'm kind of zooming in here. I'm zooming into just a little section of the mitochondria here. And what I'm showing you here is the inner mitochondrial membrane in the outer mitochondrial membrane. Okay, so in between that is the intermembrane space. Intermembrane space. Okay. On the outside, obviously, is the cytoplasm of the cell, and on the inside is the mitochondrial matrix. Okay, the mitochondria is really kind of a complex structure, right? So one thing I'll kind of focus on here, I'll put this in orange, is that within the intermembrane space is a compound or chemical called cytochrome C. That's this little bit in orange. Okay. Now this ends up becoming a pretty important signaling regulator during apoptosis. And normally, the outer membrane of the mitochondria is intact, it's not permeable to cytochrome C, so cytochrome C is held within this intermembrane space. However, there are some proteins that can actually create pores in the mitochondrial membrane that allow cytochrome C to be released from this intermembrane space to then start apoptosis. So what are these proteins? Okay. There are many proteins, but two of the key ones are known as BAX and BAC. Okay, these are essentially proteins that can kind of reversibly form pores in the membrane of this outer membrane. Right. So if there is some sort of, I'll put this in yellow, some kind of a cell death signal. Okay, this can converge on these proteins, activate them, okay? And then as a result, these proteins end up localizing to the outer membrane of the mitochondria and produces pores here and here, okay? Now those pores are permeable, importantly, to cytochrome C. So these pores that allow for cytochrome C to exit the intermembrane space, enter the cytoplasm, and then these then activate other proteins to then activate apoptosis. Okay. That's the broad context here, right? Regulating apoptosis through regulating the release of cytochrome C from the mitochondria. This is clearly an oversimplification. There's a lot more interesting detail here, but ultimately I want you to remember this. Cell death activates these proteins, BAX and BAC. They form pores in the membrane. Cytochrome C can be released. This is really what kickstarts another signaling cascade which produces programmed cell death, right? So there's our broad context, okay? So let's bring this back to BCL2L1 splicing, okay? All right. So there are two splice forms. For BCL-2L1, right? Okay, and these are going to be called, these result in two different protein isoforms that results in BCLXL and BCLXS. Okay, in this case, the L stands for long and the S stands for short. Okay, now the mechanism here is through an alternative 5' splice site. Remember back to our examples of alternative splicing, this one uses an alternative 5' splice site to produce these two splice forms, right? So let's take a look at the BCL-2L1 gene. It's actually relatively simple. Consists of 3 exons, okay? 1, 2, and 3, okay? Now the alternative splicing occurs right here in exon 2. Okay? And so there are 2 splice sites that can be used at the 5' end of this intron, right? So these are then alternative 5' splice sites. So you've got splice site, we'll call this the normal splice site right here, the 5' splice site right there, and you've got an alternative 5' splice site right there. Now, the choice between these two splice sites is what creates these two different isoforms here, right? So, if, in the case we'll do on top, we'll do the BCLXL, we'll do this one in green rather, and we'll do the BCLXS, do that one in purple, okay? So the long isoform is produced by splicing here, okay? So this produces this splice form with full exon 1, full exon 2, and full exon 3. Okay, and this includes that bit there in exon 2. Okay, this is why this is called the long isoform. So this is BCLXL. Okay, L for long. That includes this. Right? Now, alternatively, let's say we splice here as normal, but we use this alternative splice site right there, okay? This then produces a splice form that has the full exon 1, okay? A truncated exon 2, and full exon 3, right? So here's what we're missing. We're missing this chunk right there, okay? This is BCLXS. Okay, that's the short isoform. Okay, and this ends up producing 2 different proteins. Okay, now the difference here is this chunk here. This ends up coding for 63 amino acids. Okay, and we'll kind of get to why these 63 amino acids are important here in a second. Right. So those are the 2 splice forms. I'm going to go ahead and erase this, but these are the key differences. The long isoform includes this chunk. The short isoform excludes that chunk. Okay? Now these 63 amino acids turn out to be pretty functionally important for what these 2 proteins can do. Right, so key here is that BCLXL is an anti-apoptotic protein. Okay? It prevents apoptosis from happening. Whereas BCLXS has a completely opposite function. This is pro-apoptotic. Okay, so these things kind of function at this sort of critical decision point from whether the cell undergoes cell death or not, right? So let's look at kind of, we'll focus on a situation in which BCLXL is present first, right? So we'll go back to our diagram here. Here's the inner membrane. Here's the outer membrane. Okay, remember there's cytochrome C in this inner membrane space. Okay. Okay. Remember, this is the cytoplasm out here. This is the matrix. Okay, and this is the intermembrane space, or the IMS, right in here. Okay. So in this case, BCL-XL is anti-apoptotic because it actually inhibits the function of the two proteins we talked about before, BAX and BAC. So remember, now I'm going to have to put BAX and BAC in different colors here, okay, just to sort of clarify things. But remember, we've got these two proteins, BAX and BAC. And those are the ones that during cell death can actually go to this outer membrane and form pores. Well, it turns out that BCL-XL can actually inhibit each of these proteins. And it does this by actually interacting with these proteins right here. So I'm going to draw in right here a little orange chunk, okay? And what that represents are those 63 amino acids that are included in XL but excluded from XS. So these are the 63 amino acids I just talked about, okay? It turns out that these are required for interaction with BAX and BAC, okay? All right, so BCL-XL, I kind of show these inhibitory lines here, but let's erase those real quick and just show that BCL-XL can actually bind to BAX here, again requiring those 63 amino acids. Okay, and I'll flip it around here, but it can also then bind to BAC. The same idea being it's these 63 amino acids that can bind and interact. Okay? So this prevents these proteins from forming pores. So in this situation, these pores that would form don't occur, okay? Which then prevents cytochrome C from being released, which prevents the sort of whole cascade of signals that induces cell death, right? So this is how BCL-XL is anti-apoptotic, okay? So let's contrast that then with BCL-XS, right? So in this case, I'm going to kind of use the same diagram, but just kind of reset it, right? So here's our outer membrane. We still have cytochrome C in there, okay? Okay, we've got BAX and BAC here, okay? Now in this case, we're producing BCL-XS, the short isoform, okay? Now remember, this lacks those 63 amino acids. That's the key here, right? And as a result, it can't bind and interact with BAX or BAC, okay? So in this situation, this interaction can't happen. And I'll put X's here to sort of let you know that this interaction doesn't happen, which then allows BAX and BAC to go to the membrane, form pores, okay? This then allows cytochrome C to leave the intermembrane space, make it out to the cytoplasm. This can then lead to that apoptotic signaling cascade, apoptosis, right? What's also interesting about this is that BCL-XS, part of the way this changes the function is that this also actually allows BCL-XS to bind to and inhibit BCL-XL. Okay. So BCL-XS inhibits any BCL-XL that's being made. So often in this case, it isn't just a switch between you're only making one or the other. Oftentimes you can make a little bit of one splice form that can help kind of alter the function of the other one. In this case, producing BCL-XS actually inhibits any of the BCL-XL that's around. Okay, remember BCL-XL is our anti-apoptotic protein, so by inhibiting an anti-apoptotic protein, you further kind of produce this entry into apoptosis, right? So this is a key decision point in this case. And so part of the way that apoptosis is regulated is by switching the splicing of BCL-201 to produce BCL-XS instead of BCL-XL, okay? And this helps promote apoptosis, okay? Now, I'll end this by just sort of talking very broadly about what should kind of enter your mind as a biologist when you start hearing the word apoptosis and cell death, okay? You should almost automatically start thinking about one of the key disorders related to this one, which is cancer, right? Okay. Misregulation of apoptosis can also be a component of how tumor cells are formed, right? So oftentimes a tumor cell will kind of start to sort of get into its sort of tumorigenicity, and oftentimes there's a decision point of, as it starts to divide rapidly, of whether it should continue dividing or undergo cell death, okay? If the cellular mechanisms are working properly, the cell can appropriately kind of enter cell death instead of continuing to grow and divide. And this is oftentimes in the face of, say, too much DNA damage or other kinds of stressors that are causing that cell to be damaged. The appropriate response is for that cell to undergo cell death. However, some mutations in cancer cells can actually affect that decision point, such that a cell that is normally intended to go into cell death ends up surviving, right? So there are mutations, we'll say some mutations, some cancerous mutations produce too much survival instead of apoptosis, right? So in this case, there are some mutations in some cancers that are associated with this splicing point itself, right? So if you misregulate this splicing, and if the cell is supposed to produce this BCL-XS spliced form, and instead it can't and keeps producing this BCL-XL, this is our anti-apoptotic signal. If there's too much of an anti-apoptotic signal, this produces too much cell survival, and this can lead to then the irregular growth and division of a cell that should have undergone cell death, right? So there's an even broader context to kind of put this into, right? But fundamentally, what I want you to take out of this is that this alternative splicing between these two forms of the same gene produce very different effects, and that can become a key regulatory switch for a particular cell, right? Okay, those are the two videos. I will get an email out with just some general administrative announcements, things like that. Please watch these videos. Again, the content of these videos will be on exam number 2, which is Wednesday in class. Thanks.